In today's video, I'll show you how you can observe animal cells under a microscope in a simple, at-home way. Many people think this is difficult and only possible in a laboratory, because it's true that plant cells are much easier to observe. All you need is a piece of onion or a leaf, and you can immediately see the cell walls and sometimes even the movement of cytoplasm. Animal cells are usually more challenging, but there is one exception that works perfectly for learning: epithelial cells from the inside of the cheek. Let's start with what epithelial cells actually are. These are cells that cover and protect the surfaces of our body. They can be found, among other places, on the skin, in the oral cavity, nose, intestines, and lungs. Their role is to protect the body against mechanical damage, chemical substances, temperature changes, and the entry of microorganisms from the external environment. Now let's move on to preparing the slide. To collect the cells, you'll need a regular cotton swab. Gently rub it against the inside of your cheek. This way, you collect cells that naturally shed from the surface. Then transfer the material onto a clean, dry microscope slide by, lightly pressing the tip of the swab against the glass. After spreading the material, leave the slide for a few minutes until it is completely dry. The next step is staining, because unstained cells are almost invisible under the microscope. First, apply a few drops of hematoxylin to the slide and leave it for about 5 minutes. After that, gently rinse the slide with water to remove excess dye. Then perform the second staining step with eosin. Again, apply a few drops, but this time leave it on for only about 30 seconds. And then rinse the slide with water once more. The prepared slide is now ready for microscopic observation. It's best to start with the lowest magnification to easily find clusters of cells, and only then gradually increase the magnification to observe individual cells. With hematoxylin and eosin staining, you can notice that the cell nucleus stains blue or purple, while the cytoplasm appears pink. This is a very classic and commonly used staining method in histology. Of course, you can also test other dyes. For example, I tried methylene blue. Then both the cells and their nuclei appear blue, which also makes observation easier. The cells we are observing come from the inner side of the cheek and form stratified squamous epithelium. This means it consists of many layers of flattened cells. Because this epithelium is in constant contact with the external environment and exposed to various damaging factors, it renews itself very quickly. In the lowest layer, called the basal layer, there are undifferentiated cells that divide intensively. New cells gradually move upward, differentiating along the way, and eventually being shed, which is exactly why we can collect them so easily from the cheek. To sum up, observing animal cells doesn't have to be difficult or complicated. Cheek epithelial cells are an excellent material for learning. They are easy to access, simple to prepare, and very clearly visible after proper staining. If you want to see real animal cells with your own eyes, this is one of the simplest and most impressive ways to do it.